Hello everyone, welcome to today's live broadcast, Oxidative Stress and Schizophrenia, a Translational Approach. I am Judy O'Rourke of LabRoots and I'll be your moderator for today's event. We have a few important announcements before we begin. This presentation has been approved for a Category 1 Continuing Medical Education credit by Loma Linda University. If you are interested in obtaining CMEs after attending this presentation, please click on the Get Your Free CME CE Credits button in the bottom left corner of the event screen. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Professor Quang Do. Professor Kim Do is Director of the Center for Psychiatric Neuroscience at the University Hospital of Lausanne, Switzerland. Her current major interest lies in bridging basic neuroscience with problems of clinical psychiatry. Since 1999, she set up a translational research program aimed at a better understanding of the causes and mechanisms leading to schizophrenia phenotypes in order to develop markers for early diagnosis, new drug targets, as well as preventive and therapeutic measures. Building on an innovative hypothesis, she demonstrated that oxidative stress, induced among others by glutathione GSH deficit, may represent a hub on which both genetic and environmental risk factors converge during neurodevelopment, leading to the impairment of neural connectivity and synchronization and to cognitive deficits as observed in patients. In addition to her research activities, Professor Kim Do also works on professional education and public outreach towards better care and cure of major psychiatric disorders. I will now turn it over to Professor Do for her presentation.
Hi, everybody. Today I want to begin with a story with a young adolescent, 14 years old, with school problems, perturbed behavior, with 16 years old, drug abuse, begin to hear voices, 18 years old, the first psychiatric hospitalization, and from there on, 18 to 21, many crises of emergency hospitalization and failure of professional formation. This is a typical story for a young adult affected by schizophrenia, which is a brain major psychiatric disorders affecting 1 to 3% of the world population if we also include all psychotic disorders. It is a neurodevelopmental disorder with a complex interaction between genetic and environmental risk factors, with a progressive evolution and psychosis generally start during adolescence and young adulthood, which is a critical phase of psychosocial development. And schizophrenia presents a quite heterogeneous clinical picture, and the symptoms are classically divided in positive symptoms and negative symptoms on which are based the diagnostic. Positive such as hallucination, delusion. The negative symptoms which more characterized by retreat of personalities affecting language, affects, will, pleasure, and social domains. But maybe the most invalidating symptoms are the cognitive deficit, which affect all executive function. And not talking about the basic symptoms, which are maybe the more specific one for schizophrenia, affecting perception, motor, and self disorders. So these apply well for chronic phases, but not all to the early phases. Psychotic disorders also is known to evolve through various stages. So the first psychotic episode is known to be preceded by a prodromal phase with subthreshold symptoms, and treatment delay has negative impact on the outcome, increasing the relapse rate. and early detection and early intervention has become a major focus for research and service development. And Philippe Conus in our psychiatric department has designed a clinical strategy to improve this outcome in order to reduce the delay to diagnostic and also development of stage-specific treatment. And for that, we need valid biomarker for a good definition of stage for early intervention, and also we need for mechanism, to understand mechanism for having stage-specific treatment targets. So for that, we have, since some years now, developed a translational approach in which we have in parallel both the clinical branch, so working with patients, and the preclinical branch, working with animal models, and trying to have the same assessment endpoints in the two branch, from genetic, biochemical, morphology, neurophysiology, psychopathology or behavior or social functioning in the two one, and having a tight interaction between the two clinical and preclinical branch in order to develop biomarker which may be useful for early identification of patients, definition of various stages, assessment of illness progression, and monitoring of treatment efficacy, and also understand mechanism for new treatment and even maybe treatment, preventive targets. So the cartoon now from Thomas Insel highlights the main anomalies found in chronic schizophrenia, mainly the deficient myelination and the reduced, so excitatory inhibitory imbalance. And today's talk will mainly focus on the reduced interneurons activity. And the question still remain open for the earlier phase, whether these impairment could be used as early biomarkers which are needed to enhance prediction accuracy of clinical measures and to develop early intervention strategies. Now, today's talk, I would like with you to ask three questions. Could redox dysregulation be responsible for some schizophrenia phenotypes, including fast-spiking parvalbumine interneurons and myelin impairments? Could glutamate, NMDA-receptor impairment, inflammation, and dopamine overload interacting with oxidative stress be considered as a core mechanism? And could antioxidant redox modulators protect against oxidative stress and contribute to the prevention of the disease? Let's now first look at the pathophysiology of schizophrenia. Based on our own work and also from work of many labs, we propose that redox dysregulation in full interaction with NMDA-receptor hypofunction and inflammation represent one hub on which converge various causal, genetic, and environmental risk factors during the neurodevelopment and leading to structural and functional connectivity impairment, which then could be at the basis of the symptoms and dimension. In terms of genetic risk factors, so many genome-wide studies containing up to 300 patients highlighted, for example, the dopamine-related system genes. And you may have heard the newest aspect of the role of dopamine in the disease with the webinar of Professor Anthony Grace. Also, the glutamate-related genes, or the NMDA-receptor function and oxidative stress genes, or the antioxidant system-related genes, as well as the immune system-related genes. So highlighted in red are genes which has been known to be directly involved or generating oxidative stress. And now in terms of environmental risk factors, many known risk factors affecting the fetal, perinatal, or childhood period, such as inflammation, hypoxia, preeclampsia, or insults in adults have been shown to induce oxidative stress. And this is the convergence of these two risk factors may lead now that we will see to redox dysregulation, and we will see how redox dysregulation, oxidative stress, may lead to microcircuit affecting parvalbumine interneurons and macrocircuit affecting myelination. So our first spring-kept observation was the decrease of glutathione levels in cerebrospinal fluid of drug-naive patients, and later on confirmed by magnetic resonance spectroscopy in prefrontal cortex of schizophrenia patients. And searching for the cause, we have seen that the key synthesizing enzyme of glutathione, so the main endogenous cellular redox regulator and antioxidant, so these genes are associated with the disease. And most importantly, we have seen that the high-risk genotype concerning polymorphism of the key synthesizing enzyme of glutathione, the gclm, so the high-risk genotype has functional consequences. As you can see here, in fibroblasts of schizophrenia, chronic schizophrenia patients, the high-risk gclm genotypes, when challenged with oxidative stress, has lower glutathione content and also lower synthesizing enzyme activity and protein expression. And recently, leaching seen, thanks to magnetic resonance spectroscopy, so MRS, has shown now in early psychosis patients that the high-risk genotype patient also has lower glutathione levels as compared to the low-risk genotype. So, GCLC, so the glutathione high-risk genotype, can predict low brain glutathione levels. So, now at least in early psychosis patients, the glutathione levels decrease may depend on the percentage of high-risk patients in the samples. We are not the only lab who have seen oxidative stress in schizophrenia. Many studies have shown abnormal antioxidant levels and signs of oxidative stress, both in peripheral tissue and nervous tissue of schizophrenia patients, and including also copy number variation and risk gene, which are directly related to antioxidant system. So the redox balance, which is the balance between antioxidant and reactive and oxygen and nitrogen species, is quite critical in the brain, as the brain, compared to other organs, has a very high oxidative metabolism. So when the unbalance is quite severe, oxidative damage on proteins, lipids, DNA can occur and have drastic, irreversible effects. But if there is only a slight unbalance, so that kind of what we call redox dysregulation, there can be reversible oxidation of thiol residues on proteins that can have change modulations of the function and can have impact on neurotransmission, proliferation, differentiation of cells, specifically early in the development. And I want also to mention redox control is more general than oxidative stress. Indeed, it is hypothesized by Dean Jones that besides being involved in direct signaling, such as the redox signaling pathway here, antioxidant systems such as thioredoxine or glutathione can exert a modulation on other signaling systems, such as the ionic signaling system that we will see today, an example concerning NMDA-receptors, or the kinase system. And they can thus exert orthogonal control on other signaling systems, leading to a coordinated physiological response. So let's look now how redox dysregulation can lead to microcircuit impairment impacting on parvalbumine interneurons. Let me begin to show you this cartoon, which show actually the microcircuit, which is composed of a pyramidal cell sending collaterals to the basket cells, inhibitory neurons containing parvalbumine, a calcium-binding protein, which can in turn inhibit the pyramids. And the succession of excitation, inhibition, lead to oscillation at high frequency, at the gamma frequency. And these basket cells are unique in their fast spiking properties. And the microcircuit allow PVI to time and synchronize neuronal networks activity in gamma frequency, and has been shown by many, many labs to be very critical for cognitive functions. So, works of many labs, especially that of Francine Benes, David Lewis, show that PVI, as well as the NMDA-receptors, are impaired in schizophrenia, leading to a disrupted information processing and integration mediated by gamma oscillation in neuronal networks, then thus can contributing to the symptoms and cognitive deficits known in schizophrenia. Moreover, PVI are particularly vulnerable to redox imbalance, as their fast spiking activity implies enhanced oxidative metabolism, generating more ROS, and thus may need high antioxidant protection. We now ask the question, could oxidative stress play a role in PVI alteration? And or just like in our case, a glutathione deficit. So to investigate this question, we use a genetic animal model of glutathione deficit in which the key synthesizing enzyme of glutathione, the glutamate cysteine ligase, the modulatory subunits, so the GClm, are knocked down, and these mice has a quite uniform glutathione deficit across different brain regions and throughout life. And using a marker for oxidative stress, the 8-oxo-deoxyguanosine, which is a product of DNA oxidation, we first observe the presence of oxidative stress in the ventral hippocampus. Here shown is the CA3 regions in adult gclm knockout, which is not present in the wild type. And by contrast, there was no sign of oxidative stress in the dorsal hippocampus. And in parallel, Jan Carbunkel can show that the PVI immunoreactivity in the ventral hippocampus is also impaired, so is decreased, and particularly strong decrease in the CA3 and dentate gyrus, where oxidative stress, you can see here, are the most prominent. And you will know more about the role of these two regions with the webinar presentation of Professor Carol Taminga. And this impairment on PVI is not present in the dorsal hippocampus. And consistent with that morphological alteration, so we have looked also since parvalbumine interneurons are necessary for the neural synchronization in the gamma frequency range, Pascal Sterle has looked for the ability of kinase to induce local gamma oscillation in hippocampal slice. And you can see here that the gamma oscillation is quite decreased in the ventral hippocampus, while it was not affected at all in the dorsal hippocampus of knockout mice. And consistent with the PVI impairment and neural synchronization deficit in ventral hippocampus, all related behavior to ventral hippocampus, such as impaired emotion, stress, social behavior, and sensory gating, are altered in knockout mice, while the behavior related to dorsal hippocampus remains unaffected. So now we ask the question, how is the gene-environment interaction between redox and the environment? So as mentioned earlier, a severe environmental insults could lead to additional oxidative stress. So together with Pascal and Jan, we have asked the question, does an additional stress affect PVI in the anterior cingulate of knockout mice? And is there any specific period during the development when additional stress is particularly deleterious to PVI interneurons? So here you can appreciate that early life stress, increased oxidative stress, and decreased PVI in anterior cingulate of gclm knockout. And we know that environmental insults would induce an increase of dopamine release in the prefrontal cortex, and dopamine itself may also generate ROS and so H2O2 or quinones through its metabolism. Thus, to mimic environmental stress, we treated our mice with a dopamine uptake inhibitor, the GBR, which then induce an excess of extracellular dopamine in region, so with dopaminergic terminals such as the prefrontal cortex, and we treat that during juvenile, during peripuberty, 30 to 40, or during adulthood. And then assess for oxidative stress. And you can appreciate here that is, after the juvenile, peripubertal, or adult stress, there are a strong increase of oxidative stress marker, which is present in the knockout but not in the wild type, indicating that in the wild type, without the genetic risk, the upregulation of antioxidant system is quite efficient. Now, concerning PVI immunoreactivity, you can appreciate here that the childhood and the juvenile and the peripubertal stress decrease PVI, while the adult stress has no effect at all. So if I can summarize here for you, looking for PVI counts, you can see here it is the wild type, and in the knockout gclm knockout mice, there is only a decrease of PVI in the adulthood, significant. But now if we can add additional stress at the juvenile, you can see a strong decrease of PVI in knockout, not in wild type. The same when the stress is applied at peripuberty, but the adult stress has no effect at all on the knockout mice. And now, what is more interesting is that the juvenile stress, which lead to a decrease of PVI, the effect persists into adulthood, here, as you can see here, in the knockout mice. And so the question comes, could PVI impairment be reversed or prevented by antioxidant treatment? So we use N-acetyl-cysteine, which is a safe antidote for cysteine and glutathione deficiency, known as a precursor of glutathione, and itself has also an antioxidant effect. So we treat since pregnancy on NAC, and you can see here NAC can totally reverse the decrease of PVI in knockout mice after the peripubertal stress. So if I can summarize, in the mouse, a glutathione deficit, so coming from a gene, risk gene, combined with the stress, environmental, during development, leads to lasting impairment of parvalbumine neurons, of their perineuronal net. I don't have time to show you, but we have seen that also. And the neuronal synchronization in the medial prefrontal cortex. Induced during infancy and adolescence, these effects of stress are deleterious and long-lasting, while they are not at all if occurring in adulthood, and they can be completely reversed by the antioxidant NAC. We can then speculate now, translation to patient, that individuals who carry a genetic vulnerability has a risk to develop psychosis if they are exposed to environmental insults during brain development. Those insults can be infectious, traumatic, or psychosocial, and has been shown that at the origin of oxidative stress, they could induce impairment of PVI, of myelin, as we will see after, and of neural synchronization and cognitive function. So it is also tempting for us to speculate that the timing of the environmental insults impacting on at-risk individuals may determine which microcircuit or macrocircuit would be structurally or functionally affected. So depending on the neurodevelopmental stage, at the time when the gene-environmental interaction is taking place, different circuitry, so such as sensory, motor, or language, or cognition could be affected, and different could then lead to a kind of heterogeneous clinical phenotype. And this hypothesis is currently being tested in our lab. So, can redox dysregulation also impair microcircuits, so now impacting fiber tracts connecting various brain regions? So white matter abnormalities as assessed by DTI are found both in chronic and early stage of schizophrenia, and there are converging evidence also that myelin and oligodendrocyte alteration are present in schizophrenia. And we have made an intriguing observation. So thanks to multimodal imaging using MRS, so magnetic resonance spectroscopy, assessing glutathione in the medial prefrontal cortex, and diffusion spectrum imaging, assessing white matter integrity along the cingulum bundle, we see that is a correlation, both in control and patient, of glutathione with white matter integrity as assessed by fractional anisotropy here along the cingulum, suggesting that brain glutathione levels play a critical role in white matter integrity. So it is also known by work of Mark Noble's lab that the redox stage is a central modulator of the balance between proliferation and differentiation of oligodendrocyte precursor cells, which are making the myelin. And actually, he can show that the reduced states lead to more proliferation, while oxidizing status lead more to differentiation. And this is what Aline Monin has tested in vitro in oligodendrocyte precursor cell culture, and in GCLC shRNA, glutathione deficit lead to decreased proliferation, totally reversed by NAC, early differentiation, and also impaired myelin maturation, as you can see here. And this is consistent also, we can see in vivo, where glutathione deficit in GClm knockout mice lead to impairment of myelin here with myelin basic protein markers, so in vivo, in anterior cingulate at peripuberty at various layers of the cortex. So what could be the underlying mechanism? We thus ask the question whether glutathione deficit conditions, the switch of oligodendrocyte precursor from proliferation to differentiation, could be mediated by the Phin kinase pathways. Indeed, Phin, which is a non-receptor tyrosine kinase, is regulated by redox-sensitive proteins. And oxidative stress conditions lead to upregulation of Phin, upregulation of CCBL, decrease of PDGFR, and the switch from proliferation to early differentiation. And this is that Aline can show both in vitro, in oligodendrocyte precursor, this dysregulation of the Phin kinase here activity, and also in vivo in GCM knockout with the Phin mRNA expression. And the question is, can these results can be translated and validated in patients? And very interestingly, together with Margot Fournier, Aline can show that the same impairment, that means here the upregulation of finkinase activity, can be seen in patients. Actually, it is early psychosis patients in the fibroblasts, specifically the high-risk glutathione, the high-risk genotype glutathione patient. So this regulation of fin in patients can actually be predicted by our oxidative stress models. So what are the fiber tracts that could be affected by oxidative stress? So Alberto Corcoba, with Rolf Grüter and João Duarte, has developed a cutting-edge 14 Tesla diffusion tension imaging in GClm knockout mice, assessing the wide mitral integrity in the whole brain in vivo. And you can appreciate here the reduced fractional anisotropy in the fornix fimbria as well as in the anterior commissure in GClm knockout mice, and not in the wild type, so compared to the wild type. And here with the longitudinal study during development, so Alberto can find that there is a reduced fractional anisotropy already present at peripuberty and existing all along until adulthood. Now, what is the functional insight of fractional anisotropy alteration, so the FA? And interestingly, Pascal Chelet can show that this is correlated very well with reduced conduction velocity in the same fibers in the fornix as measured by electrophysiology. So the fornix fimbria is the main output of the hippocampus and very critical for cognitive function, and in this alteration is involved in memory deficit. We then ask the question, can the white matter integrity alteration of the fornix in our mice, in our model, can predict the same anomalies in patients? And this is exactly what Philip Bowman and Alexandra Krifa has looked for in early psychosis patients. There is a significant decrease of fractional anisotropy in the fornix, as well as a decrease of the hippocampal volume. And interestingly, in the patient, we can find a correlation between hippocampal volume and FA in the fornix, that reduced white matter integrity in the fornix is associated with smaller hippocampal volume, and this is present in the patient but not in the control. So, may I summarize? In the macrocircuit, myelin anomalies are found in both patients and models of oxidative stress. The finkinase dysregulation is an underlying mechanism of myelin impairment, both for models and patients. They could be as potential early markers. We have identified also impairments of the fiber tract, the fornix, important for memory, in the early phase of psychosis, correlated with hippocampal volume. And again, here we can speculate that this process may also affect many other pathways. Now, let's switch gear and go to the glutamate, NMDA receptors and inflammation. So let's first look at NMDA receptor hypofunction and interaction with redox dysregulation. Just a reminder that NMDA receptor hypofunction play a key role in schizophrenia pathophysiology, as the antagonists such as ketamine, phencyclidine induce positive, negative, and cognitive symptoms in control subject, exacerbates that in patients, and also in psychosis and hallucination has been found in patients with autoimmune NMDA receptors encephalitis. And last but not least, the deficit known in schizophrenia in auditory evoked mismatch negativity are associated with impaired NMDA receptor function. So I want now to quote very interesting, wonderful work of Jill Hartingham, who has shown that activation of synaptic but not extrasynaptic NMDA receptors boost the intrinsic antioxidant system. So namely the thioredoxine and peroxytocin pathways. But very recently, now in press in Nature Communication, Jill's lab can show also that NMDA activity play a key role in regulating the capacity of glutathione system through the direct transcriptional control of glutathione, promoting its synthesis, recycling, and utilization. And thus lead that NMDA receptor hyperactivity during development thus may lead to deleterious loss of this control. And moreover, a pioneer work of Stuart Lipton has shown that on the NMDA receptors there are redox sensitive sites in which, so on specifically on NR1 and NR2A, in which either a reducing agent such as D-thiotrietor, DTT, or glutathione can potentiate the NMDA activity. So, and also NMDA hypofunction induced either by ketamine, as shown by Margarita Behrens, or in conditional knockout of NR1, as shown by Nakazawa, leads to PVI impairment through oxidative stress. As you can see here, Marga has shown that NMDA receptor with ketamine lead to induction of cytokines, IL-6, induction of NADPH oxidase, which the main generator of ROS, and superoxide, and leading to PVI impairment. So we speculate that glutathione deficit either can act directly or can act through NMDA receptor hyperfunction, leading to PVI impairment. And this is what Pascal has looked for. He can show that glutathione deficit leads to NMDA receptor hyperfunction and long-term potentiation impairment in hippocampal slice. So now, so you can see that redox dysregulation and NMDA receptor hyperfunction can interact in synergy, creating a vicious cycle that is particularly deleterious for PVI. Now, how about inflammation and redox dysregulation? There is now emerging evidence for an increased inflammation in the brain, CSF, and blood of schizophrenia patients. And as you have already seen, there is an increase of oxidative stress in our knockout. And as you can see here in anterior cingulate at the peripuberty, so P40, as shown by Daniela Dvir has shown now also observed in the same region and same age an increase of microglial activation. And this is quite specific, as in region with no oxidative stress, such as somatosensory cortex, there is no sign of microglial activation. And more interestingly, the oxidative stress-induced microglial activation is more prominent at peripuberty than at adult stage, suggesting that neuroinflammation may be early, may can serve as early markers. Now, it is known also that there is a tight and reciprocal interaction between redox dysregulation and neuroinflammation. So ROS can induce inflammatory pathways, while microglial activation induces ROS production. Oxidative stress produces H, which is a ligand of rage, which can then induce an inflammatory response via NF-kappa B. And vice versa, neuroinflammation can increase S100 beta, also a ligand of rage, which can induce ROS via production of NADPH oxidase. So making rage, the receptor for advanced glycation end product, a potential link between the two processes. And this is what Daniela has tried to test. And here, Adanya tried to see if oxidative stress can induce rage shedding as an potential underlying mechanism. So you can see here rage can be shed or cleaved into extracellular, so a soluble form, and intranuclear rage, which can then be translocated to the nucleus and induce all the inflammatory response. And you can see here that in GCLM knockout mice, there is an increase of rage shedding as well as an increase of S100 beta, so the ligand of rage. So here again, there is a tight interaction between inflammation and redox dysregulation that could lead to a deleterious positive feed-forward effect. Now, so to summarize, redox, immune, and glutamatergic system can form a triad in which each of its elements can influence each other, and dysregulation of one system would in turn impact on the other system. We now ask the question, good models independent of redox manipulation also lead to oxidative stress? And in collaboration with Patricia O'Donnell, we then studied the developmental rat model of schizophrenia, the so-called neonatal ventral hippocampal lesion, the NVHL, in which a lesion of the ventral hippocampus has been done at P7, and all the schizophrenia phenotype, related phenotype, emerged during adolescence. And we can ask the question, could oxidative stress be one of the mechanisms underlying this delay emergence of schizophrenia phenotype? And can an antioxidant, as a NAC, can reverse it? And you can see here, Jan Kabunkel can see there is a strong, so both at juvenile and adolescence, so P61, an increase of oxidative stress marker, but in parallel, parvalbumine cells are only decreased in adolescence, but not significant in juvenile. And more interestingly, if we give NAC before the lesion, we can prevent all these alterations. So besides this morphological alteration, Patricia can show that NAC can reverse electrophysiological response, such as the D2 modulation, loss of synaptic response in medial prefrontal cortex of pyramidal cells, as well as the mismatch negativity, which is quite related to NMDA-receptor hypofunction. And more importantly, that NAC can also prevent the sensory gating, as assessed here by the pre-pulse inhibition. And now we ask the question, how about, we have now treated NAC before the lesion. How about now if we treat the animals only during adolescence? And quite interestingly, NAC can also reverse the alteration in PPI, even if the treatment is given only during adolescence. So, the oxidative stress and PVI impairment is induced by interventions which are independent of redox manipulation, suggesting that it is one of the core pathophysiological mechanisms. And oxidative stress may be one mechanism underlying the delay emergence of anomalies driven by developmental alteration, and preventing oxidative stress with NAC or excellent and another antioxidant can rescue all the morphological, physiological, and behavioral schizophrenia phenotype, thus paving the way to prophylactic treatment. Now, I just want to end, so to end with collaboration. So the very recent results done in collaboration with Takao Hensch. You may know the landmark work of Takao Hensch on the critical periods of plasticity. His lab can show that the opening and closure of critical periods are mainly depend on PVI maturation and also on the perineuronal net. This is the extracellular matrix with enwrapped PV and which protect also PV from oxidative stress, and also myelin formation are critically involved in these two processes. And this is the two processes which are precisely the factor impaired in redox dysregulation condition. So in conditional knockout of glutathione synthesizing enzyme, specifically on PVI, Morishita and Jan Kabunkal can show that there is an increase of oxidative stress and decrease of PVI and PNN. And more interestingly, they can show that in adult monoocular deprivation, in the knockout mice, the conditional knockout mice, the ocular dominance distribution continue to be shift away from the deprived eyes. And the same result you can see here in the contralateral bias index, the CBI. In the knockout mice, there are still a shift of ocular dominance plasticity beyond the peak of the natural critical period. So in redox dysregulated condition, the neocortex remain plastic even beyond the peak of the natural critical period. So suggesting that redox regulation may balance the plasticity and stability of cortical networks, and the mistimed developmental trajectory of plasticity may underline, at least in part, in the pathophysiology of schizophrenia. So now, as told you in the beginning, it is so important for us to come back to the patient. And this is what we have done in the proof of concept clinical trial together with Michael Berg to give NAC in the double-blind randomized placebo-controlled trial to 140 patients. And you can see here that NAC, given in addition to antipsychotics, can improve the negative symptoms, so symptoms which are not well treated by the antipsychotics. And also you can appreciate here already the effect can be already seen after 2 months. Here is the severity of the symptoms with NAC. And besides that, NAC can also improve the NMDA-receptor dependent mismatch negativity as well as the local synchronization. So the correction of redox dysregulation with a safe precursor for glutathione open new perspective for treatment and prevention. And this is what we try to now test with a randomized double-blind clinical trial now in first episode patient together with Philip Konus and Larry Simon. So for the perspective, we can say that maybe the intervention, if there are some, intervention should start early, possibly at the time where there are the environmental insults, and maybe with some efficient but benign treatments such as redox modulator impacting on this triad hat that I have discussed with you before. So in conclusion, redox dysregulation in full interaction with NMDA-receptor, glutamate dysfunction and neuroinflammation is a core mechanism for both micro and macro circuit alteration in schizophrenia. that in microcircuits, oxidative stress resulting from conversions of risk gene and environmental insults during development affect selectively PVI and perineuronal net in a region and time-specific manner, and early insults are more deleterious than adult ones and lead to impairment lasting in adulthood. They may shed light on potential mechanisms underlying long-lasting consequences of early environmental insults in the context of environmental risk factors for schizophrenia. In terms of macrocircuits, redox dysregulation can impair proliferation of oligodendrocyte precursor to the fin kinase pathway, leading to myelin deficits in models and predicting fin kinase dysregulation and dysconnectivity in patients. So together, micro and macrocircuit deficits lead to an imbalance of excitation inhibition and to an impaired local and distant neuronal synchrony, thus underlying cognitive behavioral anomalies in analogy to patients' phenotype. And many of these impairments can be prevented by timely administration of N-acetyl-cysteine, thus opening new perspective for stage-specific treatment and for prevention in patients. So I would like to acknowledge, and all my students and postdocs. I'm so fortunate to have so talented and motivated students and postdocs. And also would like to thank Philip Konus, the head of the Service of Psychiatry, with whom I am working now from hand in hand to build this program, and also Michel Pueno, which has been a constant support and with whom I have also started this program. I also thank all my collaborators in Lausanne, as well as Patricia O'Donnell and Takao Tench. And also thanks to all the financing agencies, especially the Synapse, the NCCR Synapse, and all the private foundation, the Foundation Avina, which are instrumental to help us to do this project. Thank you for your attention.
Thank you for that informative presentation. Before we get started on the question and answer session, I'd like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. Our first question is, would you recommend that higher levels of glutathione, cyanohydroxybutene, and cysteine sulfur-containing foods, normal foods, are incorporated into inpatient mental health children's wards and recommended to those in need in the community?
So, yes, this is quite an interesting question. The main problem with glutathione is that it doesn't cross well the blood-brain barrier. So this is why NAC has been used. But NAC has also some limiting factor. Is cross the blood-brain barrier, but also not very well. So we have to find much better redox modulators. So I cannot answer right now, but this is quite interesting. Yeah.
Next question is, extrasynaptic means?
What Jill Hardingham has shown in this, his, I think, Nature Neuroscience paper, and now also in the Nature Communication with glutathione, he has induced intrasynaptic, so he can stimulate the NMDA receptors in the synapse, but not the extrasynaptic. I think it's through treatment with 4-aminopyridine. You may have a look on this paper. So intrasynaptic versus extrasynaptic, okay? So
Next question is, what are the hypofunctions of NMDA? Is there any compensatory mechanism for NMDA hypofunction, e.g. MGLURs?
The question is there any compensatory mechanism for NMDA receptor hypofunction, for example, from the metabotropic glutamate receptors? Yes, maybe there has been work of other groups showing that hypofunction of NMDA receptors may lead to increase of so glutamate levels, and then acting on metabotropic receptors, namely the 2,3 class, can decrease the release of glutamate. Yes, there are some work on this part going on. Yeah.
Can schizophrenia be diagnosed in childhood?
This is quite challenging until now. So normally it's quite difficult to be diagnosed in childhood, but there are a cohort which is now, you may heard about, that Judith Rapoport and Nitin Gote has at the NIMH are following since long time, which is childhood onset of schizophrenia, and that has been detected in children very early. So the same sign that we see in adult schizophrenia.
Does lithium provide antioxidant benefits in this patient group?
Quite interesting question. So as far as I know, there are some work in this part, but there is not yet convincing evidence for that. So I cannot say exactly about that.
What tests for GSH function should be done and when to help prevent schizophrenia? There's a couple questions here. I'll throw them all at you. Let me know if you need them separately. So anyway, what tests for GSH function should be done and when to help prevent schizophrenia? What doses have been used in humans? Would it help to administer NAC when given immunization with mercury-based preservatives?
It's quite a lot. Now let me answer first. Okay. Let me answer first. What tests for glutathione function should be done? So now, as said in my talk, we are trying now to see if we can have some peripheral markers for glutathione deficit, but we are developing so magnetic resonance spectroscopy to see glutathione deficit in the brain. As you can see here, even the high-risk genotype can predict already the low glutathione level. But something that one may also think about is testing on the parvalbumine impairment. So we know that parvalbumine impairment leads to decrease of gamma synchronization. So that can be also, that may assesses with EEG in patients. And I know that many groups are developing and trying to see that in patients. Okay, so
could you tell me, please, whether you see any link between autoimmune disorders such as multiple sclerosis and redox state schizophrenia?
Interesting question again. As you can see, autoimmune, so autoantibody against NMDA receptors has been associated with psychosis and hallucination. and that can be treated, so the symptoms goes away with a treatment with immunodepressant. Okay, now with redox dysregulation, this is a quite interesting test. This can be, I don't know. That has now to be tested, and this is mainly that we are doing right now in our knockout mice, trying to see if with NAC we can reverse the microglial activation and also all the ray shedding that we are doing right now. But for autoimmune disorder, it's difficult to know right now. Yeah.
Have you looked at the new medication GLYX13 in respect to NR2A subunit malfunction in NMDA-R? Is your opinion might it help?
Quite interesting. What I know that the glycine, it has been now tested by, for example, the D-serine, which is acting as a co-agonist of NMDA-receptors. So as shown by Dan Javid, has quite improved the negative symptoms. But for the glycine X13 on NR2 subunit, I don't know if somebody, but it's worth to try. Yes, it's worth to try. But I suggest that it may do that. Okay, it may have some effect on reversing or at least ameliorated the NMDA receptor hypofunction.
Would you recommend adding NAC supplements to schizophrenic adult drug regimes?
So, of course, now as NAC is in the research, we cannot propose as really an, as a treatment. But what we are trying to do now is to see if NAC can improve subtypes of patients. Of course, NAC cannot be a miracle treatment, but may apply to a subtype of patient, the one which has oxidative stress or redox dysregulation or even NMDA receptor hypofunction. This is what we are trying to do. But as NAC has rare side effects, many, so around here in the Lausanne, there are many psychiatrists who has given that to adult patients, and some it is anecdotic, but some has got a quite nice improvement. But this is what I can do. But we are doing that kind of research right now.
Unfortunately, we are out of time. I want to thank the audience for their interesting questions and participation in today's event. I would also like to thank Professor Doe for her informative presentation. Today's webcast will be available for on-demand viewing for several months following. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.